Welcome back to the nervous system. The topic of this presentation is going to be graded potentials. So with the nervous system, we need a way to relay information from one neuron to another. We need the ability to roll that around probably a lot of neurons in our integration, the central nervous system, and then relay some information out to decide we're going to use a muscle and contract to get this drink of water. We're going to reach out with the nervous system to, say, some sort of hormonal tissue to release some hormones. But first we have to collect information, bring it in, think about it, decide what we're going to do, and roll that out. So each of these neurons is going to have some sort of input of information to it in the dendrites, to the cell body, and then decide if it's going to share that information downstream. If it does, it will relay down the axon across that synaptic cleft there to the next neuron, or perhaps to another neuron, and then perhaps maybe to a tissue type. So these are going to be relays, and we have to be able to talk from one to another, change that electrical signal over to a little chemical signal in that cleft, to another electrical signal, and so forth. Right? We can do this between these neurons using what's called graded potentials. We're going to collect information in dendrites along the cell body with these graded potentials. And however many times these little dings of graded potentials, which is going to be a little short-lived, localized change in that membrane potential that we have set up. Now these signals can be a whole bunch of little strong ones. They can be having the cell actually get a positive signal to go towards relaying to the next cell. We can also have a negative signal, actually make it more negative in the cell, which would take away from that cell being able to relay information. We can do this several ways. But what we can have with these graded potentials is usually going to be an upstream neuron through those little synaptic clefts talking to the cell body, or talking to the dendrites and just kind of knocking on the door, a little tap, tap. Hi, I'm here. I want you to maybe relay a signal, or hi, I'm here. We probably shouldn't relay a signal. And that can be done using different chemical signals through these synaptic clefts to tell that cell, please turn on and send a signal down your axon, or hey, I don't want you to send a signal. And it can be a negative effect on that cell and not roll down the axon. So there's lots of different types of graded potentials that we can do. And basically, like I said, this is a whole bunch of knocking of doors. Here it only shows 3 neurons talking to this one, but especially in the central nervous system, you're going to have a lot. There's going to be a lot of integration, a lot of talking. So there can be adding and subtracting of all these signals. We're going to need to reach a certain threshold of these graded potentials to allow for the other type of signal a neuron can do, that action potential along the axon. This lecture is going to focus on the graded potentials, the little touches that we get that can add up to, now let's relay a signal to the next neuron or to the tissue at the end of my axon. All right, so a little information. I just wanted to give you a visual here. Let's get some text in and start adding some words to this. Graded potentials, that's what this talk is all about. We have 2 types of changes to that set up membrane where we have a negative minus 70 millivolts inside, okay, compared to the outside. So we've set up a current capability. Now, the 2 types of things we can do is this one, the graded, where we're going to get little tickles that could possibly move in either direction and tell the cell, yes, share some information with the next cell in line. Action potentials will be another lecture. But the gradeds are these cute little, just short-lived knocks. They're very localized to that synaptic cleft and will make a minimum change in that membrane potential that, again, is short-lived, and that membrane is going to write itself back to having the minus 70 inside. Now, the stronger the stimulus we have in that localized little graded potential, the more voltage change, the farther the current will flow. And when we get a whole bunch added up on that cute little cell, it can then relay, reaching what we're going to call threshold, to kick off one of those nice long action potentials along the axon. What stimulates, what triggers that flip in that charge along the membrane is going to be using some gated channels. We use some to create that -70 inside and compared to the outside. So we have some gates then, ion channels that can open and close to either, like I mentioned, we can cause a depolarization where we're going to become less negative and potentially get to that threshold to allow for an action potential along the axon. But sometimes, if we don't want to relay a signal, the upstream neuron can cause hyperpolarization, which makes the interior of the cell even more negative and further away from allowing for that kickoff of an action potential along that axon. Graded potentials, we can give different categories. One that we can do is named according to the location and the function as well. So a couple terms here for you. You've got receptor potential, also known as generator potential. And these are graded potentials, these little short bursts, these little knocks on the door, in the receptors of sensory neurons. So that'll be one of the first signaled cells. And if we go back and look at that relay here, this is what we're talking about, that initial collection of information. Okay, and the sensory here, it's a visual nerve running through the optics to the integration system. That one has the name of receptor potential or generator potential. It's going to generate a relay of signal. Okay, graded potentials and receptors of sensory neurons. Now, to relay signal, we have that synaptic cleft. So you've got to have a cell talking to another cell, right? And we'll have a postsynaptic potential. Okay, this is going to be a term where we have a neuron graded potential. Okay, so we have to relay a signal. And that's going to be on that postsynaptic or post after that synaptic cleft. Now, I mentioned that the graded potentials are very short-lived. Let's look at a little piece of membrane. So let's say we're going to go here and we're going to look at this little region here of a cleft between that axon terminal and this little strip of dendrite. So this is our presynaptic cell, the synapse, and then this little dendrite here is the postsynaptic cell. So if we blow out that little bit, we have our cute little membrane. We know it's more negative inside, -70, compared to the outside of the cell. If that little synaptic cleft, let's say it's right here, and it does a little tickle, a little stimulation, a little knock on the door, right? This whole membrane is going to kick off. There's going to be a few receptors in that localized region that are going to open up and allow a rush in of positive, a rush out of negative to flip this region here in the cell, just to give a little localized knock on the door. Now this one, because it's causing it to become more positive interior, that's the depolarization. We're going away from the negative charge and depolarizing towards a more positive charge. Right? This is a perfect example of a graded potential, just a little localized change. So what I just showed you there is once we had those gated ion channels open because we released some sort of signal there, chemical signal that said, hey, open these doors, that depolarization will spread from one area of the membrane to the next, but just in that localized region. So that first initial tickle that I showed you will just have a little flip of the membrane. But then gated channels are going to open on the other sides as well, and we're going to have a little bit larger region that's going to flip membrane potential. You see that right here. Depolarization is going to spread. Right? More gated channels open. A larger region, that dendrite, is going to become more positive on the interior and a little bit more negative on the exterior. Opposite charges are going to attract each other, and this is going to create that local current. You see here these back arrows as we roll and have that flip of membrane potential. So it does spread like a wave, and we're going to see that with the action potential as well. It's going to run from one end of the axon to the other. Here, because we're getting tickled in the middle, it's going to roll in either direction, left and right, along this cute little dendrite. The big thing here with the graded potentials is the current flows, right? Those gated channels are open, but they quickly close, and that's going to cause that signal being more positive interior and negative on the outside to change back. All right, and if you need to refer back to the video on how we set up that graded potential, or set up the ability to have a graded potential, please do so, because that's what will happen. Once these gated channels close, that membrane is going to reset itself to have that minus 70 on the interior. So we look at graded potentials again. Little short region of tickle, okay, and it's going to be short-lived. If we look at this change in depolarization, okay, and distance along that dendrite, it's only a few millimeters where we're going to have a site of depolarization. Sites to the left and right will still have that resting membrane potential. That is the big picture of a graded potential. Short-lived, localized area. Now imagine you have a cell tickling graded potentials here saying, hey, relay the signal please to the next cell. Another dendrite might be coming in, or dendrite here having a lot of little synaptic clefts saying, oh no, don't. Another one coming in, oh yes, do. If we have enough flipping of this membrane to reach a certain threshold in this axon hillock, the very beginning of the axon, that is when we can have the next type of potential, which is the action potential. This is much different from the graded, where it's a little touch and done. The action potential we're going to see is an all or none. It is going to, once it starts, it is not going to stop until it reaches the end of the axon and allows that cell to relay a signal to the next neuron or next tissue in line. So that is all I have now on the graded potentials. Again, it is a brief, very short distance localized signal within that neuron. We're going to have a short change in that membrane potential. And again, depending on the signal relayed with that chemical signal across that cleft, we can either go into a depolarized state where we get closer to relaying an action potential, or we can push into a hyperpolarized where it actually becomes even more negative on the interior and harder for that cell to reach a threshold where it will relay signal to the next cell using its axon and its cute little terminals as well. Graded potentials are going to occur on receptors. For the sensory neurons, if they're going to collect information, that's going to be called a receptor potential or a generator potential. And graded potentials occurring in response to a neurotransmitter released from another neuron is going to be called the postsynaptic potential. Post being after the synapse, postsynaptic. All right, short and sweet on them graded potentials. See you soon on the next lecture.